UV light, ionizing radiations, and other environmental factors damage DNA, resulting in the alteration of specific bases, strand breakage, cross linkages, etc. So, all these are large scale DNA abnormalities. For such large scale DNA abnormalities, nucleotide excision repair is suitable. In this, the DNA double helix is unfolded to show the damaged section where the faulty fragment of DNA is identified. After that, an excision nuclease, also called as exonuclease, cuts the DNA on either side, upstream and downstream of the damaged DNA. The defective piece is degraded. The gap created by the nucleotide excision is repaired by DNA polymerase and DNA ligase with proper nucleotides. Defect in nucleotide excision repair mechanism leads to Xeroderma pigmentosum. So, in Greek, xeros means dry. So, xeros is equal to dry, derma is equal to skin. This condition is inherited as autosomal recessive. There are 7 Xeroderma pigmentosum genes, A to G, which code for polypeptide, which is necessary for nucleotide excision repair mechanism. Mutation in any one of these genes, it may lead to this Xeroderma pigmentosum. So, 7 subtypes of Xeroderma pigmentosum are there, each caused by the deficiency of a different polypeptide in the repair system. Enzyme deficiency is ultraviolet specific endonuclease, and the important features are individuals suffering from this condition are extremely sensitive to the light, that is, they are photosensitive. Freckles and ulcerative lesions are developed when exposed to sunlight. Coming to skin cancer, the risk of skin cancer is very high, that is about 2000-fold higher than the general population. Typically, this is seen in their first and second decades of life. In some subtypes, unrelated symptoms such as progressive neurological degeneration, developmental delays, and corneal ulcerations are also seen. The only effective treatment for this fatal disease is complete avoidance of the sunlight, and using sunscreen ointments will be beneficial.